narrowing in on sort of a one vector fits all cone applications, as you'll see. Okay, so first question, why target cones? Well, this audience, I don't need to describe that, but if you knew nothing about cones, but you saw the vasculature, the retinal vasculature of a primate, you would say, well, there's something funny going on in the middle. And that's the avascular region. Is this not on? That's the avascular region that is over the cones and over the foveal cones, and of course it's avascular, so you have the highest acuity in the foveal cones. A reason for studying cones, and this just is a little bit better anatomical view of the retina. Here, human retina, and you can see that if you look down here, a plot of cone density versus rod density right in the middle, right in the foveola, is the highest cone density, and that corresponds to this region here in the histological section. So this is the region that we're really trying to target because it's responsible for our highest acuity vision, color vision, face recognition, reading. Okay. What cone diseases are important to think about treatment for? The criteria are really pretty obvious again. We want a gene that targets foveal cones. The genetics needs to be well established. It should have a serious human phenotype, as it should have a blinding phenotype. We'll talk about one phenotype in cones that isn't a serious disease, but there's a reason for that. And the animal models for that specific cone disease need to mimic as much as they can the human disease. And obviously there needs to be a clear clinical endpoint if you really want to get into a clinical trial with cone-targeted gene therapy. So let's look at the genes that might be involved. This is, of course, a cartoon of phototransduction. And what I want to do is highlight the cone genes that can cause foveal disease. There are three that cause achromatopsia. Achromatopsia is a loss of all cone function. And it's a serious blinding disease. Patients basically have no central acuity, see with their extrafoveal photoreceptors, rods. And those genes include a minor form of achromatopsia, the alpha subunit of transducin, GNAT2 gene, and the two major forms of achromatopsia, the A3 and the B3 subunits of the cation channel. That's sort of the final step in hyperpolarization after photon absorption. So there's three genes there, and there's two more, the red and green opsin genes. If one of them is lost, it will lead to X-linked color blindness, red or green, which is the non-serious cone disease, and then a much more serious disease, blue cone monochromacy, where both red and green opsins are lost, and so you really still have no foveal function because there's no blue cones in the fovea. So this is maybe not quite as serious as achromatopsia, but it really falls into that class as well. So just a bit about delivering the vectors. At least up until a year or so ago, it was thought that you had to go subretinal with your viral vector, and in the case of anything that's not a primate, the cones are kind of spaced out amongst the rods, and so a subretinal vector needs to deliver its payload to the cones under this circumstance. So we're going to have an AAV serotype, and an AAV5 serotype, as we have found quite a few years ago now, is very efficient at transducing rods and cones. We need a cone-specific promoter, and then we'll have a specific cone cDNA for that disease that we're trying to treat. For most of what I'll say, almost all of it, we're going to be using a promoter called PR2.1, which was actually initially designed by Jeremy Nathans. He proved that this PR2.1 promoter, which is the red cone opsin promoter, human red cone opsin promoter, will in fact drive cone-specific expression in a transgenic animal. So we're using his promoter for many years now. 2.1 means the size, the length is 2.1 kilobases in length. Okay, first model that we studied was this GNAT2 model, a relatively rare form of achromatopsia, and the vector is the serotype 5, PR2.1, using the mouse GNAT2 cDNA, and that was published in 2007. So what I'm going to do is give you only a behavioral outcome for each of these animal models, not the ERGs, not the structure. Just have to assume it's there, or the referees wouldn't have accepted our publications. But the behavioral model for the mice, anyway, is this, as you know, this virtual rotating drum that has stripes that can rotate. And if they rotate to the right, the animal will move his head to the right if he can see the stripes, and he'll use only his left eye to do that. And you can then change the parameters of the stripes, so you can measure visual acuity by how wide the black and white stripes are. There'll be a point at which he can't see the stripes anymore. That's his threshold visual acuity. And of course, conversely, stripes go to the right, his head moves to the right, and he's using his right eye. I may have said right and left wrong before, but you can see what I was saying. Okay, so for GNAT2, I'm only going to show the one outcome here: visual acuity, untreated eyes, wild-type eyes in red, and the treated eye in blue, and you can see there's been an improvement to essentially wild-type visual acuity, mouse visual acuity anyway, with a very good p-value. Second model was for the A3 form of achromatopsia, one of the subunits of the cation channel. Again, serotype 5. We used a CBA, a general promoter here. We have, subsequent to this paper, we have used the PR 2.1 as well, and it works just fine. And it's using, with the mouse, CNGA3 cDNA. Again, the optokinetic apparatus, visual acuity, again, a little bit different order here, but untreated eye, treated eye, that regains visual acuity back to basically wild type with a p-value of 0.05. Okay, we have another animal model for A3 achromatopsia. This is a recent one that's not published. It's really run through Eyal Benin in Israel at Hadassah University in Jerusalem. We've made the vector for this model. The vector is, again, AV5, PR 2.1, in this case, human CNGA3, not sheep, not mouse, but human. And in this case, it's a behavioral test where the sheep has to navigate through this not so complicated maze, but sheep like other sheep. And so if you put the test animal over there, they like to go to their friends over here, if they can see it. And we'll see an untreated animal actually can't find his way through the maze, but a treated animal can. And here's the results. The untreated animal basically can't get through the maze, so there's really this is the amount of time it takes to get through the maze, but the treatment effect brings it right down to almost wild-type normal level behavior. So he can find his friends with one eye treated. Okay, the fourth model is a B3 achromatopsia model in dogs. There's also a mouse model, but I'm just showing you some of the results here. Again, serotype 5, PR 2.1, and again, human CNAG B3, not A3. And that was published a couple of years ago, 3 years ago. Again, this is, John gave a nice introduction to this. This is the earlier maze that Gus Aguirre and William Beltran constructed. I guess it was not William at that time, it was Andras Kamarmi and Gus Aguirre. But the dog has to find his way through this maze, and sort of that's what he's going to do if he can see his way with any efficiency at all. And the behavior is shown here, untreated eyes, treated eyes, wild type. So you can see that the treatment effect is really very profound, nearly again back to wild type behavior through this maze with a good p-value. Let's look now at a primate form of foveal deficiency, excellent color blindness. In this case, it's red color blindness, so it's protanopia, not a blinding disease, a condition, but it mimics. It allows us to test the idea that we can deliver a cone gene to a primate fovea. Again, AAV5, PR 2.1. In this case, the human red opsin promoter cDNA is what we're going to deliver. This is in collaboration with the Knightses, formerly at University of Wisconsin-Milwaukee, now at University of Washington. And I can't go into a lot of detail on this, but the testing apparatus, the Knightses trained the monkeys, the red colorblind monkeys, to select between 3 panels here. One of them has the test color, the others have some other color or a neutral color, and the idea is they've trained them to choose the one panel that looks different from the other 2. So this is a treated animal. He's just touched with his nose the correct panel, so he gets a little reward. He's drinking a little grape juice. So the results are shown. This is the scales are complicated. I don't need to go into it, but I just want to show you that a red colorblind monkey has basically cannot see this wavelength or this wavelength of light. It's mixed with other wavelengths. It's really on a color spectrum. And this is squirrel monkey 1 and squirrel monkey 2. They both demonstrate that they're protanopes. They don't see red well. They don't distinguish red from other colors, basically. This is pre-treatment in both of these animals with that vector, and this is post-treatment. Not quite back to baseline, which would be down about here, but you can see a big improvement in their behavioral ability to see red. They're now being able to pick out red at a much lower luminance than they were before. Okay, so actually in summary, I've shown you the 4 species and 4 genetic forms of foveal cone disease. Cones can be targeted by the right combination of AAV serotype and promoter, and can lead to behaviorally confirmed improvement in cone-mediated vision. Well, I had mentioned one other disease, blue cone monochromacy, and I haven't talked about it yet. Well, we're in the fairly early phases of it. We don't have behavioral studies. This is based on a rat BCM model that was discovered in China a few years ago. We're collaborating with them, and we have sent vector over there, and I've sent a surgeon as well to China to do these experiments, and they have subsequently done cone-mediated ERG analysis on these. I hope you can see this. This is a photopic flicker. Here is an example of a treated eye, nice flicker response, about 10 microvolts. And here is the untreated eye in the same animal, basically no response. The p-value is 0.008 over 6 treated eyes in 6 different animals. So we think that there is going to be a behavioral improvement when we can get around to doing that. It's difficult from 7,000 miles away, but we're getting there. So basically we can maybe change the 4 species and 4 genetic forms to 5 species and 5 genetic forms, if you will. So basically, what's next? What are we going to do with this kind of information? Well, 3 things. Good news is that it looks like, according to the NIH, and this is sequester proof, I'm told, that we will get this a gene therapy clinical trial initiated, the process for initiating a gene therapy clinical trial for B3 achromatopsia in April. So we'll see if that notice of award actually gets here. Secondly, since B3 and A3 achromatopsia patients have essentially identical clinical presentations, A3 patients will be discovered and characterized during the B3 natural history study. So A3 clinical trial will actually proceed in parallel with B3, and that will be separately funded through corporate support. So A3 and B3 achromatopsia are on track. And the BCM and gene therapy clinical trial is under development, and that's funded with private support. So at least three of the major cone-specific foveal diseases, I think, have a future in the clinic, at least testing in the clinic. Thank you.